So last week we talked about how all of your somatic cells have the same DNA. So if they all have the same DNA, how is it then that they differentiate? How is it that some of them turn out to be skin cells and some of them turn out to be eye cells and some of them turn out to be liver cells? Well, that is what we are talking about tonight in lecture 17, covering chapter 11, how genes are controlled. So we're going to talk about gene expression, that's the turning on and off of genes. We're also going to talk a little bit about cloning and about the genetic basis of cancer. So let's get started with the control of gene expression. So proteins interacting with DNA turn prokaryotic genes on or off in response to environmental changes. So we're talking about prokaryotes first. And there's quite a few terms that you're going to need to know. In fact, a lot of this lecture is going to be terms that you'll need to know. So gene regulation is the turning on and off of genes. And then when we talk about gene expression, that's the overall process of information flow from genes to proteins. The control of gene expression allows cells to produce specific kinds of proteins when and where they're needed. So we're not just producing all the proteins all the time. Gene expression actually allows us to only produce the specific kinds of proteins that we need where we need them. And we're going to give you an example. Our earliest understanding of gene control came from a study of E. coli. So in prokaryotes, there's what we call an operon. An operon is a cluster of genes that have related functions along with their control sequences. And with a few rare exceptions, operons only exist in prokaryotes. So E. coli, since it's a prokaryote, does have operons. And we're going to be specifically talking about the lactose operon, because when E. coli encounters lactose, all the enzymes that are needed for the metabolism of lactose are made all at once using the lactose operon. So the lactose operon, which we abbreviate as LAC, L-A-C, includes 3 adjacent lactose utilization genes, a promoter sequence, which is a site in, where RNA polymerase binds and initiates transcription of 3 lactose genes, and an operator sequence, which is where a repressor can bind and block RNA polymerase action. And don't worry, we're going to take a look at this in just a minute. Now, E. coli uses 3 enzymes to take up and start metabolizing lactose, but it only produces those 3 enzymes when lactose is present. So the question then was, if E. coli is only producing these 3 enzymes when lactose is present, how do the cells know when lactose is present? How are they able to turn those genes that code for the enzymes on and off because of the presence or absence of lactose? Now, the genes that code for these 3 enzymes are located next to each other, and they're actually regulated as a single unit, which is going to play into our answer here. So regulation of the LAC operon includes a regulatory gene, which is actually located outside of the operon, which continually codes for a repressor protein. We're going to talk about the repressor protein in just a minute. Now, in the absence of lactose, the repressor binds to the operator to prevent RNA polymerase action. So again, RNA polymerase would bind to the RNA and produce those 3 enzymes. But if lactose is absent, the repressor actually binds instead and prevents RNA polymerase from binding. Lactose inactivates the repressor, so when lactose is present, it actually will inactivate the repressor, it'll unblock the operator, and then RNA polymerase can bind to the promoter, and all 3 genes coding for those enzymes are then transcribed. So let's take a look at what that actually looks like. So if you look up here, here's our DNA, and we have our promoter, our operator, and then our 1, 2, 3 genes coding for those 3 enzymes. Now, if lactose is absent, so lactose is not present in the system, the operon is turned off. That is because we have this protein, which is a repressor, that really wants to bind to something. So if it doesn't have lactose to bind to, it's going to bind to this operator. So we now have the repressor, that's binded to the operator. So then when RNA polymerase tries to attach to the DNA here at their promoter, it gets blocked. It gets physically blocked by the repressor. It tries to bind, it gets blocked, so it just can't, which means these 3 genes are not expressed. Now, when lactose is present, however, lactose will go and bind to the repressor. Since the repressor is happy, it has its lactose, it's binding to something, it does not bind to the operator, which means that RNA polymerase can now go in and bind to DNA and transcribe these 3 genes. And then we get into translation into our 3 different enzymes down here. So there are actually 2 types of repressor-controlled operons. So in the example that we just looked at with the lac operon, that's an example of what we call an inducible operon. So an inducible operon is usually turned off, but it can be stimulated or induced by a molecule. In our example, that was lactose. The other type of repressor-controlled operon is what we call a repressible operon. An example of this is the TRP operon. And a repressible operon is normally turned on, but can be inhibited or repressed when a specific molecule, in the case of the TRP operon, that's amino acid tryptophan, when it's present in abundance. So we have the example of the lac operon, an inducible operon that's usually turned off, but can be induced or turned on by the presence of another molecule. And then we have repressible operons, which are usually turned on, but can be turned off by the presence of a specific molecule. So here's what that looks like. So over here's our inducible lac operon. You'll notice here that it's usually turned off unless lactose is present. The opposite of that is the repressible operon. So when tryptophan here is present, it's going to block it. When it's not present, we're going to be good to go. All right, so another type of operon control involves activators, proteins that turn operons on by binding to DNA and stimulating gene transcription. And activators help control a wide variety of operons. All right, so let's take a look at chromosome structure and chemical modifications, which can affect gene expression. So differentiation. So we talked about how all of your somatic cells have the same DNA, but all your cells don't turn out to be the same type of cell. Even though they have the same type of DNA, some of them can be skin cells, some of them can be tongue cells, some of them can be, pancreas cells. That's a differentiation. And it involves cell specialization in both structure and function, and is actually controlled by turning specific sets of genes on and off. So the genes that code for liver cells would be turned on in a liver cell. The genes that code for skin cells would be turned off in a liver cell. Almost all the cells in an organism contain an identical genome. That's what we were talking about in your somatic cells. All your somatic cells typically have the same DNA. But the differences between cell types, between skin cells and liver cells and pancreas cells, is therefore not due to the presence of different genes. So your skin cells don't have different genes than your liver cells. What's different is the gene expression. So all of your somatic cells have the same genes. The reason that they're different is because of gene expression. So eukaryotic chromosomes undergo multiple levels of what we call folding and coiling, which we call DNA packing. So what's going to be different here is we're not actually talking about changing the base coding or the sequence of the DNA. We're going to talk a little bit about other things we can do to DNA to change expression. So for this next part, we're actually going to be talking a little bit about epigenetics. So we're not changing the sequence of the DNA. We are doing other things to the DNA, which is going to change gene expression. We refer to that as epigenetics. So DNA packing is an example of this. So in DNA packing, nucleosomes are formed when DNA is wrapped around histone proteins. This gives the appearance of what we call beads on a string. It looks kind of like a necklace. Each nucleosome bead includes DNA and 8 histones. and stretches of DNA, which we call linkers, will join consecutive nucleosomes. Eukaryotic chromosomes can undergo multiple levels of folding and coiling. At the next level of packing, the beaded string is wrapped into a tight helical fiber. This fiber coils further into a thick supercoil, and then we have looping and folding that can further compact the DNA. We're gonna look at a picture right here. So here is our basic double helix on the left. You'll see these little purple balls are the histones that they wrap around. So we typically have DNA, you'll notice there are these little packets of DNA wrapped around 8 histones. When we have that packet of DNA wrapped around 8 histones, we refer to that as a nucleosome. And then in between these nucleosomes, in between these little packs, that DNA is called a linker. Then we get to our next level of packing, where these now start to coil up, right? So now you'll notice our nucleosomes are going to start to coil around each other. That's that tight helical fiber. When they get really squished together, we call that a supercoil. So different levels of packing. And that's going to be important for gene expression. Now notice we're not changing the sequence of DNA. You know, the A's and the T's and the C's and the G's, we're not changing that. We're just changing how that DNA is packed. But that is going to play a role in gene expression, because DNA packing actually tends to prevent gene expression. Because if you look at this picture, it's all coiled up here, then it gets packed even tighter down here. And so that packing can actually prevent the RNA polymerase and other transcription proteins from contacting the DNA. If it's all coiled up like this, RNA polymerase can't get in order to do transcription, in order to do any gene expression. So DNA packing can actually prevent gene expression. And the higher it's packed, the more densely it's packed, the less expression there is. So higher levels of packing, the more tightly coiled it is, can actually inactivate genes for the long term. Highly compacted chromatin, which is found in varying regions of interphase chromosomes, are actually generally not expressed at all. So this packing can actually prevent gene expression in large amounts of DNA. Now sometimes there's the removal of an extra methyl group that can turn on some of these genes. Inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence is called epigenetic inheritance. So I mentioned that we're not changing the sequence of the DNA, right? When we're talking about epigenetics, we're talking about other things that are affecting gene expression. So the idea of packing, the idea of removing extra methyl groups, all of those things are not changing the sequence of the DNA, but they are affecting gene expression. So that's epigenetics. And that can actually be passed on. So the inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence is called epigenetic inheritance. And these modifications can be reversed. We know that they can be, but we actually don't know how yet. So there's also something called X chromosome inactivation. So in female mammals, one of the 2 X chromosomes is chemically modified and highly compacted, and that results in it being inactivated. It can be either the maternal or paternal chromosome, which is randomly inactivated. And inactivation usually occurs early in embryonic development. And since it occurs early, all of the cells that are made afterwards, all of those cellular descendants, have the same inactivated chromosome. And we call an inactivated X chromosome a Barr body, B-A-R-R body. And tortoiseshell fur coloration in cats is actually due to inactivation of X chromosomes in heterozygous female cats. So this is what a tortoiseshell female looks like. You'll notice there's black and brown. The reason that they have that coloration is because of this. So we have 1 chromosome with the allele for orange fur, 1 chromosome with the allele for black fur. There's cell division and random chromosome inactivation. So in some of them, it is the chromosome for black that gets inactivated. So that will show up with orange for her wherever those cells are. Sometimes it will be the chromosome that coded for orange fur that gets inactivated. So therefore, whichever cells are like that will show black fur. So you get this nice little tortoise shell pattern. All right, so complex assemblies of proteins control eukaryotic transcription. So we're talking about eukaryotes here. Now, prokaryotes and eukaryotes both employ what we call regulatory proteins, activators and repressors, that can bind to specific segments of DNA, and they can either promote or block the binding of RNA polymerase, turning the transcription of genes on or off, because remember, RNA polymerase is what is helping to do our transcription. So in eukaryotes, activator proteins seem to be more important than repressors. So in multicellular eukaryotes, the default state for most genes seems to be off. So in eukaryotes, most of our genes seem to be off unless they get turned on. Now, a typical plant or animal cell needs to turn on and transcribe only a small percentage of its genes. So it's actually helpful that they're usually turned off because we only need to turn on a small percentage of them at a time. Eukaryotic RNA polymerase requires the assistance of proteins, which we call transcription factors. And transcription factors include activator proteins, which can bind to DNA sequences called enhancers and initiate gene transcription, other transcription factor proteins that interact with the bound activators, which then collectively bind as a complex at the gene's promoter. RNA polymerase then attaches to the promoter and transcription can begin. So if you take a look, here are enhancers. Over here on the left, we have our promoter here in green, and then we have our genes. So transcription factors like our activator proteins and our other proteins will bind. DNA bending proteins, RNA polymerase, all of those are going to work together. You'll notice there's a little bit of bending of DNA, but they basically all have to come together in order to start transcription. So coordinated gene expression in eukaryotes often depends on the association of specific combinations of control sequences with every gene of a particular metabolic pathway. All right, so eukaryotic RNA may be spliced in more than one way. So if you remember from last week, we talked about how we transcribe DNA to RNA, but then RNA has those exons and those introns, the non-coding and the coding sequences, and we actually will remove the non-coding sequences before the RNA leaves the nucleus in order to go do translation. And it's possible, though, that they don't all get spliced. They don't all have those non-coding regions removed in the same way. So we call that splicing, where we're taking out the non-coding regions. And there's something that's known as alternative RNA splicing. That's where different regions are spliced, different regions are taken out. So it produces different mRNAs from the same transcript. So we're going to be transcribing DNA to RNA in the same way. So that's what's called a transcript, that first piece of RNA that's created. But then we can splice it in different ways. And this results in the production of more than one polypeptide from the same gene. So in humans, more than 90% of protein coding genes appear to undergo alternate splicing. So if you take a look, here's our strand of DNA. We have our exons, we have our coding regions here, and then our non-coding regions, the introns that are in between. We are going to be making that RNA transcript from transcription. Remember, we had a cap and a tail, and then we have to remove the non-coding sequences, the introns, before it leaves the nucleus to do translation. But we don't always splice it. We don't always remove those introns in the same way. So you'll notice here, it could be spliced, and so it ends up 1, 2, 3, 5, right? We actually removed number 4 while we were splicing. Or we might end up with 1, 2, 4, 5, where we actually removed exon number 3. So sometimes exons get spliced as well when we're doing this, and that different splicing can produce different strands of mRNA. And if you look at it, these are going to be different when we do translation. So these are going to result in different polypeptides therefore resulting in different proteins. There's also regulation at later stages of gene expression. So after a eukaryotic mRNA is fully processed, so we've done our splicing, we've sent it outside of the nucleus into the cytoplasm, gene expression can still be regulated. And it can still be regulated by the breakdown of mRNA, the initiation of translation, protein activation, and protein breakdown. So if you take a look, here's our initial polypeptide that we've made from translation. It's inactive. We can actually fold it over, and so these S's are going to link together. So it's now all folded, the S's are going to link together, it's still inactive. But then sometimes something called cleavage happens where that polypeptide gets split. And when it gets split, that can actually cause it to activate. And in the case of this particular polypeptide, it creates an active form of insulin. There are non-coding RNAs that also play multiple roles in controlling gene expression. So actually only about 1.5% of the human genome codes for proteins. This is also true of many other multicellular eukaryotes. And there's another small fraction of DNA that consists of genes for ribosomal RNA and transfer RNA. There's actually quite a bit of research recently that suggests that a significant amount of the remaining genome, that the rest, remember only 1.5% codes, there's data that suggests that the remaining genome is transcribed into functioning but non-protein coding RNAs, including what we call small RNAs. So one of the important small RNAs that we'll talk about briefly here are called microRNAs or miRNAs, which can bind to complementary sequences on mRNA molecules, which either degrades the target mRNA or blocks its translation. This causes RNA interference. So those microRNAs, by binding to complementary sequences on mRNA, can cause what we call RNA interference. And in RNA interference, the use of microRNA to artificially control gene expression is done by injecting those microRNAs into a cell in order to turn off specific gene sequences. So we can actually do this on purpose. We can create specific microRNAs with a complementary sequence to what we're trying to turn off. So we can inject microRNAs into a cell, and those microRNAs are going to bind and to the RNA, and they're going to cause RNA interference. So they're actually going to turn off the specific gene that we want. So here's a picture of that. So here's our microRNAs that we've injected. Here's a protein. So the microRNA will actually come and it'll get in between the protein and the mRNA. And since it's now in between, and this has a complementary sequence to whatever gene is right here, instead of the protein here binding, the microRNA is going to bind, and it's actually going to block the protein, which can cause the mRNA to get degraded, so it can break down and it won't work, or it can just block translation. All right, so let's visualize the concept here. So multiple mechanisms regulate gene expression in eukaryotes. So there's actually multiple control points that exist where gene expression in eukaryotes can be turned on or off. They can be sped up or it can be slowed down. Although many control points are shown, only a few of them may be important for any particular protein. So I'm going to show you a whole sequence of where we can have control points. Not all of them are always used, but these are ones that exist. So your book likes to point out that the flow of genetic information from a chromosome to a protein is controlled at several points. It's kind of like the flow of water through pipes is controlled by valves. So if you take a look on the left, you're going to see these different valves which signal a control point. So right here at the very beginning, we have DNA unpacking. So we're here in the nucleus, we have our chromosome. Remember that DNA can be packed or unpacked in different densities. If it's tightly packed, that can affect gene expression, it can inhibit gene expression. So we have a control point there. Then during transcription, when we are transcribing DNA to RNA, there's a control point there. During splicing, when we are removing those non-coding regions and we're adding a cap and a tail, both of those can be control points. When the mRNA is leaving the nucleus and going into the cytoplasm, when it's flowing through the nuclear envelope, that's another control point. Sometimes it can be broken down, and if it breaks down, that's another control point. And then during translation, when we are actually translating the mRNA into a polypeptide, which then moves into a protein, that's another control point. So if any of these are turned off, if any of these don't work, it will block the rest of the process from happening. So if you imagine water flowing through this pipe, if any of these valves are turned off, the water is going to stop and none of the rest of the pipe is going to receive water. So it's the same thing here. If we turn off any of these points, nothing after it is going to happen. There's a couple more down there, active proteins, amino acids. All right, so cell signaling and waves of gene expression direct animal development. We're going to be talking about fruit flies a little bit. So early research on gene expression and embryonic development came from studies of fruit flies. So research on fruit flies and other developmental mutants, such as a mutant fly with 2 legs where its antenna should be, has led to the identification of many of the genes that program development in the normal fly. So there are things called homeotic genes, which are master control genes. They're kind of like a master key. They regulate the other genes that determine the anatomy of body parts. So there are genes that say, you know, these things up here should be antenna. Homeotic genes control all of those. They also determine which body parts will develop where in the fly. So I just mentioned we're going to be talking about a fly with legs on its head. It's because of those homeotic genes. If we change the homeotic genes, we can actually direct it to grow body parts in the wrong place. So if you take a look here, you'll notice this is a regular fly that has antenna on its head. By kind of modifying those homeotic genes, we can actually have them tell the fly to grow the cells here to grow legs instead of antenna. So we end up with a fly with legs on its head instead of antenna. All right, so we're going to look at the cloning of plants and animals briefly here. Now, cloning is actually fairly common in plants, not as common in animals. Plants actually do it themselves quite often. Plant cloning shows that differentiated cells may retain all of their genetic potential. So we've mentioned a couple of times that our somatic cells typically have the same DNA. So your skin cells, your liver cells, your pancreas cells all have the same DNA. So there's not different DNA. That's not why cells are different. The reason that cells are different is because of gene expression. So then the question is, if all the DNA is still present in all of our somatic cells, could they still express those genes that aren't actually being expressed? So in a liver cell, we have the same DNA as a skin cell, but it's the genes for liver that are being expressed. The genes for skin are not. But does that DNA still hold the potential to express the genes for skin? So if we're looking at plant cloning, that actually will tell us that yes, they do retain that potential. So let's take a look. So most cells express only a small percentage of their genes. So, liver cells express the liver genes, and skin cells express the skin genes. So if all genes are still present, but some are turned off, have the unexpressed genes become permanently disabled, or do all genes, even the unexpressed ones, retain the potential to be expressed? Ooh, I gave it away on the last slide. So one way to approach these questions, because scientists had these questions, right? We didn't know the answer yet, they had these questions. One way to approach these questions is to determine if a differentiated cell can be stimulated to generate a whole new organism. So in plants, this ability is common. So a differentiated plant cell can undergo cell division and give rise to all the tissues of an adult plant. So if you think about succulents, you can take a cutting from a succulent, you can take a small little part and you can grow a whole new succulent. We're going to talk about carrots a couple of slides from now and about how it can be done with carrots. So on a larger scale, we're going to look at this picture in just a minute. There's a technique that can be used to produce hundreds or thousands of genetically identical plants from the cells of a single plant. And in plants, this is referred to as a clone. And it's done through asexual reproduction. So you can take part of just one plant. It doesn't need any kind of mate. You just take a piece of one plant and you can grow a whole new plant from that small piece. So here's an example from a carrot. So you can actually take root cells from a carrot. So you don't take the whole carrot, you just take root cells and you can cultivate them in some kind of medium. And those cells will actually differentiate and form a whole carrot. So we didn't take cells from every part of the carrot. We just took them from the root, but those root cells can still grow an entire carrot. Another example of this is during quarantine, my brother decided to see if he could grow potatoes. And so he took just like the small pieces on the outside of the potato and he planted them in a bucket. And after like 3 months, he grew 5 very adorable quarantatoes. And he didn't put a whole potato in there, he just put a small piece, but that small piece was able to grow and differentiate into a whole new potato. So in animals, a good indication that differentiation does not have to impair a cell's genetic potential is the natural process of regeneration. So the regrowth of lost body parts. Think about a salamander. If a salamander loses a leg, it's actually able to regrow that leg. So it's missing a leg, right? Those cells are no longer there, but the cells around it are able to express the cells for leg growth and actually grow a new leg. So that tells us that the cells around it, if they're able to then express those genes, they have to still have the potential to be active. And many animals, especially among invertebrates like sea stars, are able to regenerate lost body parts because even cells that are not currently expressing all of those genes, they still have them and they have the potential to express them. All right, so we're gonna look a little more at cloning, specifically about how biologists are able to clone animals. And this is via a process that is known as nuclear transplantation. We're gonna take a look at that. So in nuclear transplantation, what happens is we actually will remove the nucleus from an egg cell. So this will be a donor cell. So this is actually, if you're familiar with the rhino project that the San Diego Zoo is doing right now, there are northern white rhinos, which are almost extinct. They're actually what we call functionally extinct. That means that they're not able to breed. They're either, you know, all male or they're all too old. For some reason, they can't breed on their own. And the northern white rhinos are actually functionally extinct right now. There's a few individuals left, but there's no breeding individuals left. So this is the process that they are trying to do. So they're actually going to be using southern white rhinos as a surrogate. So the zoo a couple years ago got some southern white rhinos in. They're housed at the safari park up in Escondido. And what they're eventually going to try to do is they're going to try to create embryos. And so, ooh. And there's a couple different processes that they can use to do this. Sorry for the OOs as I'm trying to like remember all of my different cloning examples from the zoo. But there's a couple of different ways that they can do this. One of the ways that they are looking at doing this is they have the frozen zoo. So they have DNA that's actually frozen, it's cryogenically frozen, both up at the safari park and down at the zoo. And they're going to try to see if they can create embryos out of that, if they can stimulate cell growth. What's been done before in other animals, especially a lot of hoof stock that I've seen, is they will take the egg from another animal, like they could take the egg from a southern white rhino, and they remove the genetic information from that egg, and then they implant the genetic information from another animal. So they could implant the genetic information from a northern white rhino. And so it would be the egg from a southern white rhino, but it has all the genetic information of the southern white rhino removed. So all of the DNA that would be present would be the DNA from the northern white rhino. And they've done this with a couple other animals already. And so then they could take that egg, and since it's a southern white rhino egg, even though it has northern white rhino DNA in it, they could implant that into a southern white rhino surrogate. And then, like, oh, I'm getting ahead of myself here. Right, so here's the donor cell. They implant the DNA from the other type of animal, and then they can implant it, and the resulting offspring would actually be whatever that donor DNA is. So in the example of our rhinos, a southern white rhino would give birth to a northern white rhino because all of the DNA present would actually be from a northern white rhino. So that's one of the ideas that they're exploring. They're trying to see if they're able to cultivate full embryos they can implant, or if they're going to have to do this type of nuclear transplantation. But this is something that they've been successful with before. It's been used in a couple different animals. Oh, there's the clone. Look at it. It's actually been done in a couple different animals before. So it was first done in the 1950s using frog embryos. And then you might remember Dolly the sheep, who was the first mammal to be cloned using this, this technique here. Now, it is important to note that the example that I gave with the rhinos, they wouldn't be exact clones, because the zoo actually has DNA from both males and females. And so it's possible that this is brand new science that they're trying to do. So this is actually, it's kind of a form of cloning, but not really. It's really much more advanced than that, because they have DNA from males and females. And so they're hoping to be able to figure out a way to then, combine those to make genetically different offspring. But the simple version that's been done before in other animals is basically just cloning, where they take the DNA from a donor and implant it into an egg. And so they're really only getting the DNA from one donor. So what the San Diego Zoo is doing is much more advanced than what's been done in the past. But they're hopeful that they'll be able to do it in the next 15 to 20 years. So we shall see, but that would be very exciting if they could. But that just adds another level. So it's not an exact clone they're trying to make, since there will be genetic information from a male and a female from two different organisms. But there's a similar process in the past that was true cloning, where we're just getting genetic information from one individual, implanting it into an egg that's had its genetic information removed, and then using a surrogate to give birth to the clone. So since Dolly the sheep, researchers have cloned many other mammals, including mice, cats, horses, cows, mules, pigs, rabbits, ferrets, and even dogs. There's a big fad, I've heard, in some regions for rich people to clone their favorite dog. But we'll get to the idea of nature versus nurture a little bit later. But it's the idea that your DNA doesn't control everything. A lot of your personality, your dog, is from the environment. And so even if you clone little precious Fido, the clone is not going to end up being your exact Fido, because it's not going to go through the same experiences. Conservation biologists hope that reproductive cloning can be used to restock the populations of endangered animals, like the example that I gave you with the rhinos, which again is not true cloning, since they're going to be trying to use the genetic information from two individuals. But this idea of cloning, of removing the genetic information from an egg and then inserting the genetic information from an endangered species, has been done before. And it's a good way of helping to restock the populations of endangered animals who just don't have enough individuals to repopulate on their own. So this is quite often done when populations are very small, or when for some reason we end up with mostly males or mostly females. So we're able to create clones in order to help bring their numbers back up. This is not ideal. What we've been doing in the past is not ideal, where we're just taking the genetic information from one individual. That's why the San Diego Zoo with the Rhino Project is trying to take the idea of cloning and improve upon it, because, for reasons we've discussed in the past, it is super important to have DNA from different individuals. The San Diego Zoo recognizes that. That's why they're focusing so much on taking, the idea of cloning to the next level when they are trying to repopulate the northern white rhinos. Because what we found is when we're just using the genetic information from one individual and we're creating these true clones, like has been done in the past, that these cloned animals are actually less healthy than those that arise from a fertilized egg, from those that have genetic information from different parents. So research suggests that the methylation of chromatin might be responsible for health problems in cloned animals. And researchers are currently investigating whether chromatin in a donor nucleus can be artificially rejuvenated to resemble that of a newly fertilized egg. So they're trying to, there's scientists working on different things. There's scientists working on trying to make the process of true cloning like we've done in the past with the genetic information from one individual implanted into the egg of another one. They're trying to see if that process can be made better so the clones are healthy. Then we also have projects like the San Diego Zoo that are working on a more advanced version of this to actually include the genetic information of two individuals. So essentially they're going to be artificially creating a fertilized egg, which would be even better. So fingers crossed, let's all hope that it works. There's also the idea of therapeutic cloning, which can help produce stem cells with great medical potential. So you've probably heard of stem cells. It's been pretty popular recently. A blastocyte can provide embryonic stem cells, ES cells, which can differentiate into an embryo to give rise to all the specialized cell types of the body. So what's special about stem cells is that they are able to differentiate into any type of cell that we want. And they can divide indefinitely when they're grown in a laboratory culture. Now, when the goal is to produce embryonic stem cells to use in therapeutic treatments, this process is called therapeutic cloning. So the idea here is that we're able to take stem cells from a blastocyte and we're able to culture them, and then we can culture them to turn into different types of cells. So we can tell them to turn into blood cells or nerve cells or heart cells or any other types of cells that we need. And in therapeutic cloning, we're going to be stimulating those stem cells to produce something specific. So say somebody has a blood disorder, we would actually be able to take their own stem cells and stimulate them into blood cells. And this is important because we can do blood transfusions, but blood transfusions are not perfect. There's always the threat that your body's going to reject the donor blood. It's the same thing if we're talking about organs, like if somebody's heart is failing and we can maybe get them a donor heart, but it often doesn't work as well, or sometimes it can be rejected. And so if we're able to get stem cells from that same person, we could then tell those stem cells to grow into a new heart. And we could basically create someone a brand new heart from their own stem cells, which would reduce all of those risks. And so that's something we've had some success with. There's still a lot of work to be done on therapeutic cloning, but it's a really big field of medical research right now because it has a lot of potential. So the adult body also has stem cells. We hear a lot about when babies are born that parents are paying to have their umbilical cord saved to get stem cells out of the umbilical cord. But the adult body also has stem cells which serve to replace non-reproducing specialized cells as needed. But because adult stem cells are farther along the road to differentiation, they can give rise to only a few related types of cells. So that's why you see a lot of parents saving stem cells from their babies, because the earlier that you get them, the more able to differentiate they are. Basically, the later you wait, the less types of differentiation they're able to do, the fewer types of cells they're able to turn into. All right, so we are on to cancer. So we are going to talk a little bit about cancer right now, and then we should be covering cancer a little more in your lab. So cancer results from mutations in genes that control cell division. So cancer is actually kind of like runaway cell division. And cancer is actually a set of many different diseases. Cancer is not just one disease. You've probably heard of different types of cancer. They're different diseases, which control the mechanisms that normally limit cellular growth. And so our body only has a set amount of skin cells, and then your body says, okay, we have enough. It has a set amount of liver cells, and then your body knows, like, hey, the liver's complete, you're fine. Like, don't just keep growing out of control. What cancer does is it actually messes with that. It messes with the mechanism that says, like, whoa, slow down. Like, we have enough, you can stop, or, we almost have enough, you can slow down with the reproduction of these cells. Cancer stops that process. And so cells will grow out of control. And scientists have learned that such malfunction is often due to changes in gene expression. So the genes that a cancer-causing virus inserts into a host can make the cell cancerous. So such a gene is called an oncogen, from the Greek onko for tumor. Over the last century, researchers have identified a number of viruses that harbor cancer-causing genes. One of the examples is HPV, which can be transmitted through sexual contact, and it's actually associated with several types of cancer, usually cervical cancer. So mutations in 2 types of genes can cause cancer. So in those oncogens that we just talked about, there are proto-oncogens, which are normal genes that promote cell division. Mutations to those proto-oncogens create cancer-causing oncogens that then stimulate cell division. So where proto-oncogens they're normal genes, they promote cell division at a normal rate, there's still those different mechanisms that can say slow down, they can say stop. What happens in cancerous oncogens is that these mutations to the proto-oncogens will create these oncogens that stimulate cell division uncontrollably. So instead of those mechanisms saying slow down and wait, that doesn't happen, and those cells just keep dividing and they 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 just keep dividing, which you can imagine causes a whole slew of problems. There's also tumor suppressor genes. So tumor suppressor genes normally inhibit cell division or function in the repair of DNA damage. Mutations, though, can inactivate the genes and allow uncontrolled division to occur. All right, so you should now be able to describe and compare the regulatory mechanisms of the lac operon, the trip operon, and operons using activators. Explain how selective gene expression yields a variety of cell types in multicellular eukaryotes. Explain how DNA is packaged into chromosomes. Explain tortoiseshell cats. How do we get tortoiseshell cats? Explain how eukaryotic gene expression is controlled. Describe the process and significance of alternate DNA splicing. Describe the significance of microRNA molecules. Explain how mRNA breakdown, initiation of translation, protein activation, and protein breakdown can help regulate gene expression. Explain how nuclear transplantation can be used to clone animals. Think about Dolly the sheep, or if we're thinking even more ambitiously, possibly a similar thing being used for the rhinos. Describe those practical applications. And then explain briefly how viruses, proto-oncogens, and tumor suppressor genes can each contribute to cancer. So that is the end of this lecture, and I will see you again soon.